No, time to go. Yes.
So thank you all very much for making yourself available for this Soil CRC Communities of Practice workshop on soil biology. And welcome to Justine and Anders and Lucas for making themselves available to contribute to this workshop. I really appreciate it. I'll just remind you to pop yourselves on mute just so that keep our background noise nice and quiet, but please feel free to raise your hand or use the reactions, use the chat box so that you can have chats along the way, and I can ask questions of the presenters, but also feel free to ask questions along the way as well. We'd like to make it interactive. Take notes. Hopefully you've all got your soil CRC pens there, so take some notes along the way. Write down your questions and any queries and things like that you have. Just a bit of housekeeping. We'll go till scheduled till about 2:30, so we'll try to keep as much time as possible. Apologies for the delayed start. And just wanted to mention to you all about the Extension Oz platform that the Communities of Practice is looking like we're going to embark on using. I've sent you an email about that. If you do have any feedback about using that, what that might look like or what it might be for your organisation, because there is some requirements for your organisation to sign a form to say that they're happy to participate. Feel free to give me a call and we can have a chat about it, or send me an email with some feedback if you like. So just wanted to mention that because we're looking like we'll try and roll that out in the next week or so. So there you go. And next Wednesday at the same time, we'll be covering soil carbon. So I just thought I'd pop that little reminder in there. So without any further ado, I will introduce Justine to kick us off. And she'll give us a good background in soil biology. I've sent Justine all the information and queries and priorities you had around soil biology, and we were just having a chat and saying, you know, it's a pretty big topic, but I'm sure Justine will be able to break it down for us. And like I say, if you've got specific burning questions, then now's our opportunity to address those and go from there. So thanks very much, Justine. Over to you.
Okie dokie. Now, is everyone able to see that presentation?
Yes.
And you can hear me?
Yes.
Excellent. Thanks for having me. I'm a soil scientist with New South Wales DPI, and my background is in ecology. So I came to soil biology through that stream, and so learned more about soils around as a habitat for organisms. So I've been working in understanding, communicating soil biology since 2001. So with the guidance of Felicity, and I've put this together to give you background about understanding soil biology, what we can do to manage it and what we can do to monitor. So let me just introduce you to the soil organisms. And this is a way we can classify some of these organisms by size. You can see down the bottom axis is a size scale. And right at the very, very smallest is the bacteria. So single cell organism. And then you've got fungi, which range from all throughout those sizes. They've got those hyphal threads. You've got nematodes, which are multicellular, but they're a very simple worm, a protozoa, single cell organism. And then you've got this mesofauna group, which are examples like the mites and Collembola, these springtails. And then you've got these larger organisms like the slaters and amphipods up to the very large organisms, so spiders and snails. So that's just a way of grouping some of these organisms by size. So what we want then to know about these organisms is what their role is in soil, and especially in terms of agriculture, and because these are the drivers of the soil biological function in the soil. And they're very important and main function that organisms are involved in is decomposition of organic matter. And it's this process of decomposing organic matter which then drives nutrient cycling. So they're responsible for breaking down any organic matter in the soil. And so that is like old roots, old insects, decomposed sort of stubble, leaves, and any sort of additions. And they convert that to a plant available form, so organic nitrogen into available nitrogen and phosphorus. And so that is a critical process that occurs in the soil. And in this process, there is carbon storage and turnover. So some of those processes create a very stable form of carbon, and that is stored and very hard to decompose. And other carbon is very easy to decompose, and it drives and fuels that cycle because the organic matter is the food for these microbes. I'll go into that a bit further on in the next few slides. So also they're responsible for soil structure arrangement, and there's a couple of different ways that they create spaces within the soils, which things like earthworms create burrows, and you've got all the organisms living in that very surface soil, mixing, creating aggregates, so groups of particles together, and then creating some soil structure, and they're very involved in that. Also, they're involved in suppressing soil-borne diseases. You get a very diverse and populous sort of groups of organisms, then they're very capable of sort of competing out or even antagonizing against other sort of other organisms that come in. So depending on the effectiveness of those native soil organisms. They also can transform some potentially harmful elements and compounds. So some are things like herbicides, fungicides, and some insecticides. Use some of those compounds as food source, and they can transform them into compounds that are less toxic. So what I want to do now is introduce you to the groups of organisms and their role that they play in the soil. And so I'm going to start with the microbes, so bacteria and fungi. So bacteria, their whole purpose is to decompose organic matter, and that's like plant roots, insects, litter, mulch, and they release plant available nutrients, as I said. So they convert organic nitrogen and organic phosphorus into available nitrogen and phosphorus. And they do this with enzymes, and that's sometimes inside the bacterial cell and sometimes outside. And so these enzymes convert these minerals, nitrates, and then available in the soil solution for uptake by plant roots. They also secrete some glycoproteins and gums, which then stick together their contact with these surfaces of contact of the roots and contact of the soil particles, and start building up what's called soil aggregates. So all these particles sticking together, making it less able to be broken down by water. And then there's a whole group of fungi, and Anders will go into this in a lot more detail, so I won't touch too much on fungi. So those hyphal threads, they decompose organic matter too, but they can also decompose lignin and wood. So you'll see those often in forests, and also then in agriculture that has forest trees like macadamias and avocado. So hyphae are very important in those kind of systems. And they can penetrate into all the large pieces of organic matter and also living plant roots. They wrap their threads around these aggregates, and that also then improves soil structure. So they groups of soil particles stuck together with organic matter and bacteria, and the threads wrapping around them protect them from breaking apart when they get wet. And fungi can form mutually beneficial relationships with plants. So they can be very specific. So this one species of fungi can live in the roots of one species of plant, or they can be generalists, and they things like mycorrhizae live in the plant roots and access extra water and phosphorus for the plant, while the fungi gets food from the plant. So mutually beneficial. And then you've got a group called the protozoa, and they're a single cell organism, and they consume bacteria and fungi and organic matter as well. And they're a little bit more mobile, and they run around in the system and chomp up, then regulating those processes. So here's a group that is up next in the soil food web called the microfauna and the mesofauna. Okay, so a very large group of organisms are the nematodes in this group. They're very small, very hard to see from the naked eye, worm-like organisms. They're estimated to be 40,000 or more species, and that 80% of the individual animals on Earth are nematodes. They're very abundant and very diverse, and they're literally everywhere on the planet and inside us. So these group of organisms eat everything. There are bacterial feeding nematodes, fungal feeding nematodes, protozoan feeding nematodes, root feeding nematodes, nematodes that eat other nematodes. So they're carnivorous, and they will very specific mouth parts, which then we can look at to determine what kind of things they eat, and we can learn a lot about the population of organisms by looking at the nematodes. And then there are a whole range of other organisms like the springtails, mites, slaters, diplura, amphipods. These are tiny creatures and insects and other groups that you can see with the naked eye, living in the soil surface and in that very close to the surface, especially in the litter, and they shred organic matter. They break up and chew leaf litter, and then that increases the area that microbes have access to. So in this process, these organisms are actually regulators of microbial activity, because if they're not there and they're not shredding, then the microbes will run out of food if the surfaces aren't abundant. So they're predators and regulators of microbial populations, and depending on the agricultural system, they'll be prevalent or they might be quite absent. So just a quick look at where they are. Most biological activities in the top 30, but that's very general. Some sites will be in the top 2 centimetres and some will be a lot further down, but it's very much the surface, and there's many reasons for that, and oxygen is one of them, moisture and carbon often they're concentrated in that top centimetres, and that's where a lot of plant roots occur as well. So, but interestingly, and things that we often forget, is most microbes live in the thin film of water around soil particles and plant roots. So they're actually water-based organisms, they're aquatic. And so they move in a tiny, tiny thin film of water. And so here's a diagram, which here's a plant root here blown up. You've got root hairs, and you've got what's called this rhizosphere, so the area around the root where all of these organisms are very abundant and very stratified. So they're very much found in this area here. So those are the organisms. So what then controls the organisms themselves and their activity? So one of the very important controllers or regulators is the soil type, like its texture, like from very, very coarse sand, which has lots of pore sizes, to very, very tight clays, which are very tightly bound and more compact. So pore size is a big important factor, and it's how then related to how much water can get in, and then how hard it is for organisms to move through. So soil type and texture is very critical, and this will put some of the limits on some of the activity and some of the organisms that you will find. Moisture is critical. Without water, you will not get organisms. And so moisture is related to soil type and how much water can infiltrate, and then season and climate. So moisture is very critical for activity and organisms' abundance. Organic matter, so this is carbon, nitrogen, phosphorus. So organic matter is microbe food. And so without food, you won't get any microbes. And so I've got something called this carbon complexity. So it's like if you've got a smorgasbord versus just pasta. You've got a range of carbon molecules, you'll get a range of organisms that will decompose that organic matter. And if you've just got pasta, you're just going to get all of the pasta eating organisms. And so carbon complexity, which is like how different rates of decomposition and different sort of chemical molecules are attached, it's important for the regulating of the organisms and their activity. Also, the rate of replenishment. A lot of agricultural processes slowly reduce organic matter, not all, but you've got crop, when you've got crop taken off, goes to market, you then are losing organic matter because all of that now doesn't go back to the soil. So the rate of replenishment of organic matter really sort of drives the activity. Oxygen and aeration. Most sort of organisms are aerobic and so need oxygen to live. Anoxic conditions select then very different organisms. So there are organisms that live in anoxic conditions and they have different processes and use different energy sources. So that's another reason why microbes are in the surface soil. So we continue on and we look at temperature, another regulator. So season, of course, in the summer when it's hot, that's going to have a big impact on activity or the location of organisms. Some can move, so they can move lower in the profile, like earthworms. Surface exposure is going to affect the temperature and sort of daily fluctuations. pH. It's a fairly wide range of pH that organisms, this is a very big generalisation, and different species will have very different ranges. But if you get optimum near 7, especially bacteria, you're going to be in areas that are quite comfortable for these organisms. Now, fungi can tolerate a lot more acidic environments, so their range might be a little different and a bit lower. And disturbance. So the frequency of disturbance has shown, if it's very frequent disturbance, you're going to get less fungi because the threads get broken up all the time and so they don't persist. So you're going to get organisms that can cope with that high disturbance. And the magnitude of disturbance, if it's completely, soils are tipped upside down, then you're going to get all those large organisms that live lower down, exposed to the surface, then that's going to have a big impact, again, with depth. So a combination of those disturbance patterns will select for the groups of organisms that can cope with that situation. And plant chemicals. So root exudates, which is chemicals that the roots move through the roots out into the soil that the plants exude. And this is actually sometimes bacterial and fungal food. They use that to attract some organisms. And there is lots of research looking at what the signalling molecules are that plants actually can say, as much as they can say, that they might be needing a particular element, and they might be able to, with finding if they are using chemical signals to attract those organisms. So that's a new and evolving area of research. So here is a typical example of where microbial activity is driven by soil moisture and soil type. So this is soil moisture on the x-axis, water holding capacity, and this is an activity of microbes in carbon dioxide, so respiration. And all of these are different soils. So you can see as moisture goes up, you've got activity going up until it gets too wet and it starts going down. So different soil types will have very different moisture curves relating to activity. And if we look at the relationship between organic carbon in the soil and microbial activity, then the examples, when you increase the organic carbon, this is the available organic carbon, you've got a relationship with increasing activity with increasing soil carbon. So given those kind of principles, how can we manage our soil biology in the different systems? So if we look at moisture, look at our system, what can we do with making sure we have enough moisture in our soil? And one possibility is a soil cover, which can reduce evaporation losses. So that could be mulch, or it could be cover crops. Another area, you could incorporate organic amendments which have a capacity to hold more water within the soil profile. Organic matter, again, quality and quantity. You want to keep increasing organic matter over time. And so what we could do is increase plant biomass. So that means fertilising plants so that we have a large biomass, which then all of that biomass gets returned to the soil and increases organic matter. You have stubble or trash in some systems that will either decompose back into the soil, or mulch can be brought in from outside to add to the soil if it's been lost. Compost is a very diverse and high quality organic matter addition. You've got a range of carbon molecules that are very easily decomposed and very persistent carbon in lots of compost. Manures, you're also getting some fertiliser effect if they're higher in nitrogen or phosphorus. If you convert cropping to pasture, that increases organic matter temporarily. You want diverse plant species, can increase the different organic matter and the quality. You have different plant species decomposed into the soil. You could use rotations, and all you might be able to do is reduce your losses, and so that is maintaining your organic matter quantity in the soil. So thinking about aeration, we might think about deep-rooted plants, something which got a nice big taproot which goes down and penetrates some compacted soils. Maybe occasional tillage is an option. There are mechanical aerators that some of the horticulture people use to create like turf aerators, like in golf courses, to create little pockets where they can get a lot of air into the soil. Organic amendments also improve aeration, and things like no heavy equipment in wet clay soils to reduce all those pore sizes. So temperature, you can't really do too much about the temperature except for what you've got on the soil surface. So if it's trying to, say if it's really hot and dry, you want to protect the soil surface, you might be able to have a cover crop or a trash blanket, or if it's too cold, that's another issue about temperature. pH, so if it's very acid and you want to increase that, you can use lime, or if it's very alkaline, sulfur. Organic amendments also has been shown to move pH towards neutral. Also, the pH, you've got to depend on the crop system and also the plant requirements for acidity and its pH range. And also the bacteria to fungi ratio, you'll find in tree crops like macadamias are more likely to have more fungi than a broadacre wheat crop. And disturbance, so there might not be too much you can do in the system, but maybe there is different frequency of tillage, and so we'll just think about the processes go on and depending on your crop. There's also potential for adding soil organisms, and there is some research going on decades which have shown some beneficial additions things like rhizobium. Rhizobium bacteria are essential in nodules for legumes, and so a lot of seeds for legumes are coated with rhizobium bacteria so that they are present as the seed sprouts and grows, and you have those bacteria then able to fix nitrogen from the atmosphere. Mycorrhizae. There are some crops around the world that people add mycorrhizae to, which depending on the host specificity. So if you've got research that shows one particular mycorrhizae is very beneficial for a crop species, then that can be added. And there are a multitude of range of new biological products on the market that claim to add soil organisms to the soil, and there's less research and understanding about what these do. So here's the example of rhizobium in nodules on a legume. Here's trichossoil. Soil. This is a I was about to say trichogramma, which is an entomological species. Trichoderma. It's a fungus, and it is sprayed in the soil to reduce other funguses, especially in onions, and these have been researched for specific crops. Farm chemicals. Now we know we add herbicides, insecticides, and fungicides, and a lot of those interactions with biology are unknown, and so that is an area of interest and new research. So I'll talk a little bit about that. Okay, so now we've got some ideas about what we might do. How do you monitor and see if it's actually happening and you're improving your soil biology or increasing? So it depends. You've got to ask yourself some questions. What's your goal? Are you monitoring? Are you diagnosing a problem? Are you wanting to do some comparisons? Do you need some specific information for a decision support tool? Because you can't just go out to the paddock and say, I want to know more about my soil biology. Now that you know how complex it is, we need to then have tools which answer your specific questions. And then what's your time frame? This week, this season, this year, or it's going to be a continuous monitoring? Because these organisms are so responsive to their environment, when you take the sample is critical, and what extrapolations you use for that information. There are a lot of limitations in assessing and monitoring soil biology. There are no standard methods. There are many methods that we use, that's correct, but there's no 1 or 2 or 3 or 4 methods that are now accepted around Australia or around the world as very effective at giving you accurate and representative information about soil biology. And there's also no specific numbers that will tell you whether your number is good, bad, or indifferent. And so there's a lot of work on this in this area, but I've got to say there's some limitations. And there's also, to date, no direct relationship with plant productivity. So, and this too will be a focus of soil future research. So given that, let me just explain a few of the soil biology testing methods that are used. And some of these are in the field, and some of these will be like for growers to do, or sort of extension activities and ways of bringing soil biology to life and making it visible. You can count and you can sample and count organisms in the field that you can see. So you can count earthworms, mites, calembola, dung beetles, or percent nodulation in your legumes. And that can be a very engaging process with a group of farmers. what we're finding a lot interest in is looking at the effective function or activity of soil biology in the field. And looking at that function of decomposition, there are a couple of methods that we use. One is called the calico strip or cotton strip test, and it essentially is unbleached calico inserted into the soil vertically and left in the soil for an appropriate amount of time given the season and the location and the climate. And then it is brought out and you can see decomposition where there's great big holes. You can see patterns on the calico which have fungi and also all sorts of activity on that. Related to that is a global sort of program called Soil Your Undies. I don't know how many people have heard of that, but you buy a pair of men's white cotton undies and you insert them horizontally at 5 centimetres and leave it 2 months and come out and take a picture of your undies, and everyone laughs. And it's you put your picture of your undies on social media, and it's a global endeavour to visualise decomposition in your soil. Also litter bags. Litter bags are like a mesh, like fry screen little bag, and we put either litter or other sort of organic material into the bag, and then we insert that into the soil, dig that up and weigh or look at the decomposition. So respiration, there's a kit that's been developed and has 20 years of research that is looking at assessing the carbon dioxide that comes out of a soil sample, and it's very easy. They've developed a guide with a gel paddle that goes into a jar, and it turns a different colour after a amount of time, which they have a colour chart like a pH field kit, which tells you the carbon dioxide level and how active your soil is. And there are a lot of people using that as a tool for extension, looking at respiration. There are a couple of indirect measurements. We can look at percent ground cover. We can take the spade and dig out and have a look at root volume and distribution, and even just do an Emerson dispersion test to put a aggregate in a petri dish in water and see how much it disperses. So if you get no dispersion, you've got your aggregates are highly stable, and if it's all just falls apart, then you have very unstable and all the particles fall apart, so there's no soil glues, and it's related to soil type as well. But yeah, you've got no soil bacteria and fungi sort of gluing those together. And so here's some examples of those. Here's calico, and here's a calico strip which we put into the ground. As you get a lot of decomposition and holes, and you get different colours, and it's very visual. Here is the Solvita sort of a respiration kit, and just a plastic jar that comes in a kit. Here's a paddle, and it has a gel insert which turns a different colour depending on the carbon dioxide. Here's some undies. So they go in and you get your undies and they're very decomposed. And here are some litter bags. Okay, this is where a lot of research has been happening, is the soil biology testing and taking a sample and bring it back to the lab and seeing if we can understand more about biological, both the organisms in there and the activity. So there are different methods looking at the numbers of organisms in there or just presence and their species. So bacteria and fungi, there are old school plate counts, which grow up on an agar plate. They don't get all of the species, so they only get some species. You can look under the microscope and look at the bacteria and fungi there, and it also has issues with getting all the species and accuracy. We don't know all the species. And there are other sort of methods using the fatty acids in these organisms to count them, and also DNA and other molecular methods to see what species are in there. We can check against a database and then say we have this many species. For nematodes, a lot of the work done is microscopy. They look at the mouth parts and you assign them to like a bacterial feeder or fungal feeder or a omnivore, and then you can assign them their functional group. Or there is actually molecular methods now which you can determine all the different nematodes, and that's building too. Look at percent root infection of mycorrhizae, and Anders will talk more about that. And this is not a numbers, but this is the biomass of carbon that is due to the microbes. So this is the proportion of the carbon in the soil that the body of bacteria and fungi. So it gives you an estimate of their contribution to the soil biomass carbon. There's a lot of work in looking at assessing soil activity, which is their function. And so one of the groups of tests is enzyme activity, so looking at the actual process that happens in the soil, irrespective of whether it's bacterial or fungal. And they can be the specific individual enzymes that convert one chemical to another, or they could be general. Things like beta-glucosidase and phosphatases to look at the phosphorus conversion. Fluorescein diacetate, and that's a test that I've used. It's a general overall microbial activity estimate, and I find that quite useful. There is further you can go with the cotton strip test and look at decomposition. You can bring the soil, the cotton strip test back to the lab and actually measure the force required to tear the calico. And that gives you an actual quantitative estimate of the decomposition. Respiration. Now, it's substrate-induced respiration, or SIR. So you add a substrate to your soil and then you measure the amount of carbon dioxide coming off. So the next slide will have a bit more about that. That's quite a powerful tool. There's potentially mineralizable nitrogen, and that's incubation to see how much nitrogen gets converted in known conditions over a certain amount of time. And that company Salvita has an estimate of potentially available organic nitrogen pool. So that's something you can do on the bench, and a farmer could do that. That's paddles again. So there's a lot of work being done in looking at the diversity or organisms in the soil sample. And I'm not going to go into any of these, but there's very much all of the genetic information that is in the soil. You don't need to know what organism they're coming from. And so that will tell you how many different types of organisms there are. Community analysis, like I said about nematodes. There's been so much work done on the different groups of nematodes and their proportions that there's a group in Brisbane that can have a soil sample, look at the nematodes, and will tell you about the soil that it comes from, and there's excess fertilizer or it's degraded, or and so they have built up over many years a sort of database and other soil traits, so that they can now be quite confident in looking at nematode community analysis and telling you a lot about what that soil is like. We can get some information about the microbial function and the diversity of that. So this is micro-res plate here, and like I talked about the substrate-induced respiration where you added a substrate. Well, this system sets up, you can add a multitude of substrates and you do it in one test. So here's some soil, you add your different substrate, different carbon source, and you get carbon dioxide, and it turns this gel a different color, and you can measure the color change. And so that gives you quite a lot of information about different carbon sources and the soil's capacity to use that. And then you can get into genetic functional diversity, where you're actually looking at the genes responsible for a specific function and looking at those and with high throughput. And so you're getting thousands and thousands of samples through, you're getting information about how much of that function can happen. Again, there's still some indirect tests that can be quite useful, and carbon fractions is always going to be useful, especially the total organic carbon value. That's very useful. And this labile carbon fraction, so the carbon that is easily available for decomposition for organisms. And there are a couple of methods that we use in Australia and worldwide. We can look at the disease organisms in our soil. We can send a sample and they can analyse using DNA and then work out what disease organisms are prevalent in the soil, and that's predicted B. And there's other associated analyses that group is doing. And you can set up rhizo box or root studies, and that's this here. So you can see plants and roots, but we've got a clear plastic thing so we can understand some and samples very specific locations. And here is a tiny weeny little DNA assessment sort of equipment. Sequencer, that's it. Yeah. It's just, it blows my mind how small that is. So just, I've got a couple of examples of studies that have been done recently by the group in Wollongbar and elsewhere, looking at effects of some of the inputs on soil function. So glyphosate was one question that was often came up. And so this study looked at compared glyphosate and Roundup. So the actual chemical you got in the container with its active ingredient, and from a range of rates. They used substrate-induced respiration to assess that, and enzymes which looked at carbon, nitrogen, sulphur and phosphorus cycling. And they looked at 3 different Australian soils over a 27-day period in the lab. And so they found that the label rate, there was very little effect on respiration or the enzymes. At the higher rates, the sandy soil was most affected. And they found that the enzyme test that they were using was not as sensitive as substrate-induced respiration. And that Roundup affected soil function more than the glyphosate did. And that's because of the surfactant that is in Roundup, which helps it enter all of the areas it needs to. And just one last one, the effects of some herbicides on soil function. They compared 7 herbicides, 1 fungicide, and there's the list there. The tebuconazole was a fungicide. They used 8 separate enzyme tests, and they looked at mineral nitrogen in 5 Australian soils at recommended rate and 5 times the recommended rate over 28 days. And they found that all the herbicides at recommended rate had little effect except atrazine. So atrazine reduced the soil function. At 5 times the rate, 2,4-D and sulfuron methyl reduced the mineral nitrogen levels across multiple soil types. So these ones at those high rates did affect soil function. And nitrogen transformation methods were more sensitive than enzyme assays. So we're starting to understand a bit more about which tests we can do for research. So I'll just summarise, soil biological testing is quite complex, and there are no optimum, minimum, maximum range numbers for each crop, climate and soil type, unfortunately, just not yet. But they do have value in comparisons and monitoring over time or answering specific questions. And it's likely that a suite of mixed indicators will emerge as useful for management decisions. We're getting there. And keep involved and keep on the cutting edge and keep getting dirty. And that's it.
That was great. Thanks, Justine. Fantastic. So So has anyone got any questions? One thing just about the monitoring, I remember talking to someone about getting a soil sample for biological testing, and just understanding that you might get a test results for sort of like the entire, a measurement of all bacteria or all fungi, but not necessarily whether they're active or not. So do you have any comment about that, Justine, so if you do send a sample away to a lab and then you get your results back, I guess understanding what they're actually measuring. Are they measuring everything that's in the soil, but not necessarily what's active in the soil? And can they give you an active measure?
I suppose if they explain what their method is, and if they are doing total, and how they're doing total, and if they're doing total and active.
Can I butt in and help out there as well? So certainly if you're looking at the functions in the soil, Justine went through them quite nicely, around nitrogen transformation. That's an active measurement, and it doesn't rely on specific microbes, but it could be a suite of microbes that result in that particular function, i.e. conversion of ammonia into nitrate. Then there are some of the DNA methods, and I think Anders will probably talk a bit about some of the DNA methodologies. Some of those include sequencing and metabarcoding, and there are options within that to look at functional genes as well, and both active and I guess passive communities. So one of the activities that Soil CRC is currently doing is we're putting together I'll call it a fact sheet. It won't be a paper, but a fact sheet on the types of soil biological measurements that Soil CRC projects are currently using. And a lot of these are actually based on work that we've both done in DPI and some of our other partner organisations, but also very much aligned with the Soil Health Institute. And they're, I guess, an equivalent in the United States. So the Soil Health Institute and the USDA work quite closely, and they've got a very nice suite of tests around soil biology, and they're developing some I think sort of Justine mentioned the issue of the holy grail, what do the numbers mean? They're starting to get, for their particular systems, some numbers, some ranges, that might indicate healthy soil, marginal soil, less resilient soil. So we're doing something very similar, and that should be out in the next few weeks, and will be available on the CRC webpage.
I forgot to end with this slide.
Oh, great.
So on the video later, you can have a look at those, and there's a Soil CRC project that's literally looking at the indicators of soil health. And so from that, you would imagine some interesting and useful measures will come out of that.
Unfortunately, I think that particular project has been cancelled, Justine.
Okay.
Because that was run through DPI, and their staff were unfortunately let go. But there is a couple of replacement options happening as part of that. So yeah, there is definitely activity within the CRC. A lot of activity in that field.
Thanks, Lucas. There was a question about microbiometers. Do you have an opinion about that, Justine?
I don't know anything about microbiometers. No.
And do you have anything you'd like to say about that?
Sorry about that, Felicity. Only from the point of view that I understand that they're imported from the United States, so that they are used in the States and they're available in Australia. I haven't used one, but I'm very tempted to purchase one to have a look at what it can do for us. Until you actually play with those things yourself, you don't really know. But they cost about, I've done a costing on them, they're $300 for the kit. That does 10 samples, and then a replacement kit's $220 for another 10 samples. So, it is reasonably expensive. It does take about 10 to 15 minutes to do each test. So that's something I'll just look into a bit further, because that's something that George and I are looking at with our PED project, to look at what things that we can utilise to do that. Otherwise, it could be a lot cheaper to use our undies.
And what does that particular piece of kit or package measure? Does it state what it measures?
Yeah, I'll just flick back to the notes on that. Won't be a second, Luke.
Thank you.
Does anybody else have any other questions for Justine or Anne's?
Okay.
Sorry, I'll just quickly, you go, Anne, if you're ready.
Okay, so it's a soil health kit, 10 tests, total microbiology in your soil, compost or compost tea, so it'll do that, as well as it's a biomass tea, reliable biomass test. So that's basically all it's saying there. I'm just going to have to get a bit more information, dig a bit deeper, but that's just the basic information on the site. But I think it just shows presence, absence.
To me, it sounds like a burst respiration test, Justine. What do you reckon? And they've then done a calculation on CO2 equals a certain biomass activity in the soil or compost. It sounds to me that's what it is.
Could be.
Like the
tell.
Solvita test, yeah.
So do you think that's, I could send you the details or the link on it, would that be worth purchasing, like to give it a go?
I reckon if you can afford $300, it's probably worth purchasing and giving it a go. Yeah, why not?
Yep.
Can't guarantee that we'll know what the results mean, but but you can give it a go.
I'm sure George and I'll come up with something, won't we, George?
Probably like a lot of those things, you have to calibrate it and all that sort of stuff. But yeah, like you say, give it a go. Thanks so much, Justine. That was a fantastic introduction and certainly gave us a bit to think about. I've got a few questions, so I might just pop them in the chat. Justine, if you wouldn't mind having a bit of a look at those and giving your opinion one way or the other. But I'd like to introduce Anders Klassen, who has also kindly joined us this afternoon. And Anders has been doing some work in rhizospheres. So Anne gave us a bit of an introduction to rhizospheres at our last meeting. So Anders, thank you very much and welcome.
Hello everybody, I will just share my screen. How are we looking?
Great.
Cool. So I've got about a year left on my PhD. I work under Lucas here in the audience, along with Mick Rose, Terry Rose at SCU, and Catherine Knock at SCU. So I've got 4 supervisors. So I'm a fungal specialist, and most of my work focuses on mycorrhizal fungi and other root-associated endophytes. And I'm using sugarcane as a model crop. It's a particularly useful model crop to use as an agronomic grass. We can draw a lot of inferences from studying mycorrhizal symbiosis in sugarcane that we just wouldn't get from model plants. Justine's provided quite a comprehensive overview of the toolkits that are available for us to look at soil microbiology. It can be a little bit dizzying and disorienting trying to figure out which is the best tool to use, how to integrate that data together. So I'd like to share some methodological and applied work that I've done for my PhD. I think it's a good way forward. It's practical, and I think it's achievable in a very straightforward research workflow. You may have heard a lot about mycorrhizal symbiosis, the promise and the peril. And there's not much use in thinking about symbiosis in our crops now if we don't understand where it began and where it came from. So welcome to the mycorrhizal world. This is Earth as it was about 500 million years ago. The fungi have ushered the plants onto the land, and there are no tall plants on the landscape. These tall structures that you see are actually some of the largest and most enigmatic fungi that ever lived, and they're the progenitors of mycorrhiza. They're about 10 meters tall. We have their fossils all over the world. The fungi that we're interested in are invisible here, and they exist in the proto-roots of these small plants on the ground. This is before the evolution of wood. Nothing has gotten very large yet. Roots are just starting to evolve, and with roots are evolving the mycorrhizal fungi. And so this is how the symbiosis works at a very stripped back and basic level. In situations where plants are under nutrient limitation, host roots will release exudates or chemoattractants called strigalactones into the rhizospheric space. And these substances will cause fungal spores in the rhizosphere to germinate. They'll follow the concentration gradient of those root exudates, make contact with the root, and at the root surface, they'll form a pre-symbiotic structure, and there they undergo a molecular handshake. There's a lot of crosstalk and biochemical signaling between the symbionts. The fungus is permitted entry into the root and makes its way through the root cortex and into the plant cells of the root itself. And they form in those cells highly branched structures with an enormous surface area. And it's across that interface that all this bidirectional nutrient exchange occurs. So the plant obtains mineral nutrients from the soil, and the fungus obtains photosynthates in the form of carbohydrates and lipids. Now, mycorrhizal symbiosis, probably the most studied symbiosis of the natural world, isn't one thing. It takes a number of forms, and it has a number of different dynamic definitions. The form of the fungal symbiosis itself takes two major morphological forms. One is the arum type and one is the Paris type. And the definition of each one, arum type mycorrhizal symbioses form branch or tree-like structures called arbustles. But just as common, if not more common in our agricultural systems, mycorrhizal symbiosis can take this other form, the Paris type. So in the Paris type mycorrhizal symbiosis, the fungus coils itself inside the root cells to create a similar broad surface area. And it's across that interface that nutrients are exchanged. Why this is important is because the same fungus can take either form or indeed can take any number of forms across the spectrum. So all of those intermediate morphological subtypes in between those extremes are possible. And each of those forms will affect the rate and extent of symbiotic development in the crop. So how do we get to here? Mycorrhiza have been with plants through every conceivable biome over geological time. They've made their way across every environment and through every scenario right into the heart of our intensely cultivated agricultural systems. I use sugarcane as a model crop. Sugarcane is one of the world's most productive and economically significant crops. We use sugar to fuel our bodies. We use ethanol to fuel our vehicles. And like any fuel, sugarcane is in high demand and production is intensifying all over the world. Sugarcane produces more above ground biomass than almost any other crop on earth. And on this basis, we think it's important to study mycorrhizal symbiosis in sugarcane because small improvements to productivity at the plant level in a system like sugarcane with the kind of planting density that we encounter and the amount of biomass produced means that we can have actually quite significant whole system consequences. Typically what we hear about mycorrhiza, whether you're reading about them in the media or even in textbooks, even with what you learn at university, is that the symbiosis is largely mutualistic and beneficial. I would argue that that's not the case. Be that as it may, here are some of the ostensible benefits afforded by mycorrhizal symbiosis in our crops. The symbiosis augments the sugarcane root system, so it extends the capacity of roots to obtain mineral nutrients from soil. And mycorrhizal filaments or hyphae are extremely fine, and they can make it into all those pore spaces and parts of the soil where roots can just simply not penetrate. Hyphal filaments are extremely good at translocating nutrients. That's what they've evolved to do. And but the benefits of mycorrhiza are not necessarily limited to nutritive benefits. There are a number of non-nutritive benefits or indirect impacts of mycorrhizal symbiosis. Mycorrhiza suck carbon essentially from the atmosphere through plants as the conduit and deliver that carbon into the soil in the form of root exudates. And it's those exudates that tend to foster these microbial communities in the rhizosphere. Additionally to that, mycorrhiza and other root-associated endophytes have been shown to have pathogen suppressive effects. They do this through a number of mechanisms. They do it through competitive exclusion, just by virtue of the fact that they're occupying the roots and they're taking up that space. They also release substances which actually can suppress pathogenic microbes, and they can also interfere or modify plant signaling mechanisms to prime the plant defense responses and enhance the pathogen suppressive effects through that mechanism. And more general benefits that Justine touched on include improvements to soil structure and ultimately downstream enhancement of soil carbon sequestration. Looking at mycorrhiza through mycorrhiza as a lens to look at other soil microorganisms is very helpful for my work, and looking at sugarcane as a model system to look at other intensely cultivated crops is also very beneficial. Sugarcane's from the Poaceae. It's a highly responsive agronomic grass, and typically to study mycorrhiza, we'll use non-mycorrhizal model plants such as Arabidopsis or Metacargo, which is mycorrhizal, and we'll try and tease out mechanistic effects in these model plant species, but it's very hard to draw generalizations from those plants and bring those conclusions into the field. So I think studying sugarcane in the field is putting us ahead of the game. Sugarcane is repeatedly cultivated over several years from the same root mass through a process called returning. And so the root mass in sugarcane crops is allowed to mature over several years. By the same token, those mycorrhizal networks associated with those root systems are allowed to mature over several years. So I'm actually able to study mycorrhiza as those fungal networks ramify and become more and more complex over time. Because sugarcane is returned, and because mycorrhizal symbiosis is contingent on carbon for nutrient exchange, we also can tease out some pretty cool effects at harvest. So when you have sudden above ground biomass removal, and that source of carbon is suddenly removed from the fungus, we see some very interesting phenomena take place. Because a break or a cover crop might not occur for several years, we see that a long-term monoculture allows us to really look at mycorrhizal effects under the spotlight. In an annual crop where you might be tilling the soil every year and a break crop might go in more frequently, you're adding a lot more complexity to the plant dimension of the symbiosis. Sugarcane is typically a long-term monoculture, grown for several years at a time before a break goes in. And so you're really simplifying the plant dimension of the symbiosis, and we're reducing the variables so that we can really see those mycorrhizal effects in sharp relief. Mycorrhiza, fungi, and root-associated endophytes are replete throughout sugarcane systems, as they are with many other Australian crops. And it's surprising just how quickly the symbiosis takes hold of the roots. It can happen over the course of about 10 days, and the roots completely transform. This on the right here, we have colonized sugarcane roots in symbiosis with two groups of fungi, mycorrhizal fungi, and a group we call dark septate endophytes. And what's happened is essentially this root has, its control has been seized of this root. So These fungi actively suppress the root's own ability to acquire nutrients through its direct uptake pathway. Not only will they knock out root hairs and occupy all those parts of the root where the root would normally absorb nutrients of its own accord, but these fungi actively release substances that suppress the root's ability to obtain nutrients on its own. And it's by this mechanism that the fungus can actually foister itself upon the plant. And the more dependent the plant becomes on the fungus, the less ability the plant has to acquire nutrients of its own accord, and a positive feedback keeps the symbiosis in perpetuity. As I mentioned in the last slide, I study not only mycorrhizal fungi, but another group of fungi known as dark septate endophytes. They get that name because they're melanized, they contain melanin, and it's thought that this dematiaceous substance, it's darkly pigmented, that it helps these fungi tolerate pretty extreme environmental scenarios. And these two fungi are absolutely everywhere in sugarcane systems. They are thought to be in a tripartite symbiosis, which is a symbiosis with 3 players. Mycorrhizal fungi are masters at nutrient translocation. Dark septate endophytes are particularly good at the solubilization of unavailable nutrients. And so, understandably, the plant will accommodate both fungi. The dark septate endophytes will solubilize the nutrients, and the mycorrhiza will translocate them to the root. Both fungi spend time inside the root, part of their time outside the root, and so they occupy a dual niche. We find them together, and so I quantify them together. They've got a large, not only ecological, but also a functional overlap in these systems. Getting right down close to the fungi is extremely important. There's a lot that our genetics tools just can't tell us. There are no molecular proxies for a lot of the things that we can observe under the microscope. And in recent years, being so enamored of the new molecular tools that have come online, it's tempting to take the most modern molecular toolkit and employ it, but without the foreknowledge of what actually goes on in the root system itself and how things like DNA amounts might translate to fungal biovolumes is not so straightforward. It's important to remember that filamentous fungi, of which the mycorrhiza are an important member, don't grow by cell division. They grow by hyphal extension. They're composed of filaments, so they're a modular and a networked organism. And so they'll extend from one end. And one of the implications of this is that many of the filamentous fungi don't have a defined number of nuclei per amount of biomass. This is very problematic for our molecular diagnostics. It's a hard problem, and it's one that I've come up against in my PhD. Mycorrhizal fungi, as I mentioned earlier, will produce nutrient transfer structures such as arbustles or hyphal coils. Different points in the crop cycle, those fungi will move from forming active nutrient transfer interfaces to structures that are more associated with storage and survival, such as spores, which contain many nuclei, and vesicles, which are associated with long-term storage for fungi and contain glycogen and lipids. Likewise, the dark septate endophytes form a number of different structures, less of them, but in different parts of the root cortex. It's not clear what these structures do, and very little is known about these fungi, despite them being cosmopolitan and they're everywhere we look. And very little is known about their functional significance, and so it's important now to kind of incorporate these kind of endophytes into our mycorrhizal studies, particularly for groups like the DSE, which perform mycorrhizal functions. And on that basis, I think it's important to expand our definition of mycorrhiza from a molecular one, using just genetics, to a definition that's more functional and encompasses all those fungal root endophytes that are non-pathogenic, but that perform mycorrhizal functions as well. An important case in point is that for crops that are considered largely non-mycorrhizal, lupins, brassicas, etc., these non-mycorrhizal host plants will accommodate a spectrum of root-associated fungal endophytes, which are not mycorrhiza in the traditional sense, but which will perform mycorrhizal functions just the same. For decades now, this has been the standard way to assess mycorrhizal fungi. It's important that we go over some of the methodological approaches to mycorrhizal assessment in crops, because the value of the data is contingent on how well we understand the limitations of the tools that we use to assess fungi within roots. And so for a long time, colonization assessments of roots have been stock and trade of mycology in crops, and it's a laborious and it's a very involved process. It takes a lot of time, and to do it properly, you need to essentially be a specialist with some taxonomic knowledge. You need to be able to disambiguate the different fungi that are occurring in the root, understand the difference between groups of fungi which have the same kind of morphological structures, which are not mycorrhizal fungi. And it can get quite complex at times, and nevertheless, it's been what we've had at hand. And before the sequencing revolution and all the molecular tools came online, we only really had recourse to this, and it still forms a very fundamental aspect of research in agronomy and whatnot. And essentially you quantify the amount of fungi that are colonizing a root. The assumption is that the more fungi you have, the higher the effect. And this is not the case. And mycologists know this. It's kind of the elephant in the room, that more is not better. We know from an abundance of evidence that the surface area of the fungi accommodating a root is not the limiting factor in nutrient transfer. So indeed, a host plant that has less colonization can actually be acquiring more nutrients via the mycorrhizal pathway than a plant that has more colonization with a less effective fungal symbiont in play. Indeed, a lot of colonization of the roots of crops may be a consequence of underperforming fungi in those systems. So yeah, what we need molecular tools to supplement this process. But if you take the fungal root colonization process to the very end, I spent 2 years doing 12,000 observations in sugarcane crops. For my master's degree, it was a long and arduous process, but we got some very useful data out of it. When you follow colonization through a crop, what you can see is when that colonization rises and falls, when it's at its highest and when it's at its lowest. So to walk you through what some of this data means, we have site 1 here on the left, which is a second return crop, site 2 on the right, which is a first return crop. We are measuring in the top 2 squares, total colonization by mycorrhiza and dark septate endophytes. And you can see at site 1, by the second return, the dark septate endophytes have been almost knocked out. We also see that, by about 9 to 10 weeks, we're reaching a peak in colonization. So that gives us a good indication of when we might want to apply molecular diagnostics to capture the maximum amount of fungal metabolic activity taking place in the crop. Total colonization is only half the picture. What you want to do is break it down into its constituent functional components. So in the second 2 squares at the bottom here, the total root colonization data for mycorrhiza has been broken down into arbustles, hyphae, spores, and vesicles. Now this is important because we're not so much interested in fungal hyphae in the roots of our plants per se. What we're interested in is arbustles. We're interested in the proliferation of these nutrient transfer interfaces, because that's where the money is. The production of spores and vesicles stays relatively low just until the end of the crop, where it just starts to pick up, and that gives us an indication that the fungi is actually not getting much from the crop, and so it's shifted its lifestyle habit into storage and survival mode, producing spores and vesicles and laying wait for a new delivery of photosynthate from the host plant. All right, so the bottom line of that project was that we were able to identify critical phases in crop phenology, or as crops approach physiological maturity, we're able to identify here optimal sampling windows for different purposes. So we have detailed data for all these weeks in harvest dates at the bottom here. This tillering window is really when you want to sample your sugarcane crop. This will be slightly different for other crops, but I don't assume it will be enormously different. Right before a crop goes into, say, an exponential growth phase, you're looking at the maximum amount of photosynthate delivery into the rhizosphere by the host plant. In sugarcane, it's particularly important to sample mycorrhiza at crop maturity, because those fungi that you can detect in the root system of the crop at maturity are the most likely contenders for the fungi that will be colonizing your subsequent return crop. So here we're trying to identify parts in the phases in the crop cycle where we might be able to get in and exploit some of these processes, make the fungus jump through hoops for us, and really ultimately understand how we might manipulate the abundance and diversity of mycorrhizal fungi and other root-associated endophytes in these systems to not only maximize production, but to also benefit our sustainability outcomes. Just when I finished this master's project, this came across my desk at the university, and Predictor B had expanded its toolkit to incorporate mycorrhizal fungi. So the value proposition of this tool, which is run out of South Australia's Molecular Diagnostics Lab at SARDI, is that by applying a number of mycorrhizal specific assays, you can detect the amount of fungal DNA in the soil. And by doing so, you can establish thresholds which would be important for, say, cereals in long fallow disorder, where crops are grown after a period where nothing has been grown on the land, and the mycorrhizal fungi inhabiting the soil have no host plant, and so their populations reduce below a certain threshold, and you'll suffer a yield penalty when you plant. Or if you've got something like canola stubble in the ground, which is not too conducive to mycorrhizal symbiosis, and which may also result in yield penalty. So I thought that was a good opportunity to take this tool and kind of hack it for research purposes, take it into sugarcane, not use it as a pre-planting diagnostic, but use it in crop, in plant during development. Some of you are probably very familiar with this process. This is how this kind of sampling works. It's important to demonstrate because it's very straightforward, it's very rapid, it's very cost effective. And I think it's yielding pretty interesting results from a research perspective. And already I think it's given me some preliminary data, which has highlighted some important questions, not just with the limitation of the tools, but also with their potential for research downstream. So a number of soil cores are just taken off the shoulder of each row, and then you homogenize those cores into a single sample. That sample is sent off to South Australia, and the DNA there is extracted and analyzed using the available assays. You can request nematode assays, or you can stack assays together. And you can build up a pretty good picture of soil health apropos of the amount of DNA that's detected. How that translates to the functional significance of the organisms or the threat level of pathogens is still being worked out. But it's, in one sense, it's a useful way in to understanding what organisms may be present and in what amounts. It's very handy from a mycological perspective, because just for an additional $20 per sample, SARDI will send me back DNA that's essentially left over from their process that hasn't been analyzed. I can take that sample and then I forward that to an organization, AGRF, the Australian Genomics Research Facility, who undertake a process called meta barcoding. And it's as simple as sending off the DNA, and then they'll tell you which organisms are present in the soil specifically. Why that's important is because Predictor will give you this kind of breakdown of which organisms are present in which amounts. So by applying Predictor, there are 6 available assays, and each assay contains a different group of fungi. They're not all species level either. Some of them are phyla or taxa, or the family taxa, genera, and there's not really any functional relationship between the fungi in each assay. And so that's where really meta barcoding comes in, because meta barcoding, that additional step that I'm employing in my research, is actually going to pick out which fungi from each assay are present and operational within a given system. So I have the microscopic data is obviously undertaken at every step of the way as we accrue all of this, and as we go through and we collect fungal DNA from roots and fungal DNA from soil, we can compare what we detect in roots to what we detect in soil. Now, many of you might be familiar with just detecting soil health, and the correct assumption to make in terms of fungal symbiosis is that the fungi that are inhabiting the roots of a crop are always a subset of the soil population. But the soil population is not indicative necessarily of the fungi that will be operational in your crop and which are of economic significance. Some data to demonstrate this phenomenon. This is taken from CRC. This is the kind of data that Predictor B will kick out, just put in a simple graph form. Along the x-axis here, you have the assays. There's 6 of them: AMF A, B, D, E, A2, and C2. As demonstrated, each of those contains those fungi, and these are the amounts that the test can detect. What's important to notice here is that from soil on the left, AMFD is the dominant group in the soil. When we analyze roots concomitantly, the dominant group of fungi in the soil is not detected in the roots whatsoever. So we don't care about this group of fungi. It's essentially latent, meaning that it's laying wait for a compatible host, but it's not yet active. It's quiescent, which means it's sleeping for an indeterminate amount of time, or it could be relic DNA, meaning that it's been detected, but it's not associated with a living organism. However, the DNA has not degraded to the point where it has evaded detection. So this is a key finding of my work, is that there's no telling from soil tests of mycorrhizal fungi which group will be dominant in the roots. AMFA2 here is the group that we're interested in, and this is subdominant in the soil. So AMFA2 is the group of fungi that we will look at, that we will analyze to identify which species is present in the roots. We identify the fungi that are present within the roots. We confirm that with the microscopy, so we provide multiple points of analysis and cross-referencing. And then that then becomes a candidate for further research. So it's that fungus that you can then take into the glasshouse and compare with other fungi from other sites where this is happening as well. We can run performance assays on those fungi, identify which ones are outperforming others, and then we can start to think about inoculation in a more meaningful sense. It's not limited to one site. We doubled the variables here by using twice the cover crop treatments. And it's at a different site. This one's at Woodburn, same thing, AMFD, dominant in the soil. AMFD is not detected in roots whatsoever. This time it wasn't AMFA2 that was dominant in roots, it was AMFE that was dominant in roots. So we have a different site. On the one hand, we have the same phenomenon. And on the other hand, we've also identified that at a different site with likely a different variety under different environmental conditions, has exerted selection pressure on a different group of fungi from the soil reservoir. So in this way, we're using the plant itself to select fungi of interest for us. Also of interest here, you can see that lupins inside roots have had a suppressive effect on the mycorrhiza, which is consistent with the literature, and it's cool to see that in actual real life. Moving forward from here, this kind of work and this kind of multi-method approach forms a new basis for establishing the mycorrhizal status of crops. And I think that by incorporating all of these different approaches, we can get a really solid understanding for the industry, and it's a strategy that could be employed across multiple industries, even on different continents. That by collecting these data from, say, all 6 major cane growing regions in Australia, we can then look at the differences between each region, and we can line that up with all the data we have from the fact that Australian sugarcane is growing more or less in a straight line down the east coast, forming what we would call a latitudinal agroecological gradient, which is essentially a declension of microclimates down the east coast. And it will be very interesting for me to see how the differences in the mycorrhizal ecology interpolate with that underlying physicochemical and climate data. I'd like to emphasize finally that you can't really do this kind of work on your own. I'm not a one-man show. It's a concerted effort. I've had a wonderful group of supervisors who have been with me since 2015, essentially. Everyone has their specialty. So mycology is a very particular thing that I'm into the terminology and all the mechanistic stuff to do with that. But it's really only by virtue of having a team behind you, someone who can specialize in genetics, someone in agronomy. You need a couple of guys on soils because soil's massive. And together we were able to paint this picture, and it's been extremely rewarding. And I look forward to moving forward. Thank you for your attention.
Thanks, Anders. That was fascinating. I can see that you're really passionate about your work, and that your knowledge is deep. And you certainly, well, made it clear for me. I've learned a lot. I didn't know much before, but I feel well versed now. So thank you very much. Has anyone got any specific questions or comments for Anders or Justine while they're here? Richard, have you been involved in this work in the sugarcane industry?
Not to this extent, no. And that it is being conducted up this way in various places, but not nowhere near to the extent that Anders has gone through, or even Lucas and that. So yeah, it's all we've heard about it, and that be good to get and have a bit of a play with it shortly. Yeah, so I imagine you're seeing some of these effects that Anders has been talking about, and that's why he's looking. Yep. Yeah, and that it's a big ball that we've got to look at. That's for sure. Yeah, and Brendan, I think you also made some comments about doing some AMF testing. So predict-to-be test, is that due to some of the fallowing issues that you've got up north as well?
Yeah, so probably the basis of our work, similar to what Anders just said, particularly looking at it from a long fallow disorder point of view, but also a real issue came from the Liverpool Plains down where George is based, where the growers obviously in a fairly diverse cropping system down there, growing canola and in summer crops, particularly sorghum, dryland cotton and mung beans, so VM dependent crops back into that canola stubble. Some real issues with crop performance back into that system or back into that situation. So we're in the process right now of setting up some fairly extensive trials, one particularly at Liverpool Plains at Malali there, George, where we got a grower last year who very kindly planted alternating strips of wheat, canola and fallow. So effectively creating a different AMF population for us with the intention of planting a number of summer crops crops this coming summer, hopefully being sorghum, cotton and mung beans, back into those effectively different AMF populations, and then looking at different treatments that we could possibly try and impose, which growers effectively, or I suppose practically, could apply to try and overcome the impact of that low AMF. So I'd be interested to talk to Anders possibly later on and get some ideas of what we could be possibly doing there. Obviously looking at growing some cover crops, which we have already planted, being barley just as a cover, and we've certainly increased AMF populations there. Looking at applying deep phosphorus to try and overcome obviously the lack of phosphorus, and also using seed treatments or liquid and furrow injection products such as Endofuse from Sumitomo, whether they have any impact or not. But other than that, certainly a huge issue is obviously the long fallows that we have experienced over the last 3 or 4 years due to drought, particularly further western areas out around Walgett, where guys, as you say, have had 6 or 7 years fallow, and what impact that's going to have when they finally do plant another crop due to long fallow disorder. So looking at cover cropping, multi-species being just straight cereals versus mixtures of legumes or mixtures of cereals and legumes together, and what impact that could have.
Mm-hmm.
Yeah, good. Okay, thanks, Brendan. Yeah, it'd be interesting to see how that goes.
So another question, Anders, sorry,
yeah.
is to say we certainly do a lot of Predictor B soil sampling.
Yeah.
But when you mentioned about the root analysis, did I miss that, or where are you sending that to get analysed?
So with all the issues you've outlined with the long fallow disorder that's going on, obviously my data is preliminary, and I'm trying to accrue more evidence to support the things conveyed in this presentation.
Yep.
My contention would be that it's not the total numbers of AMF aren't the critical factor. It's your plant genome combination. Your lower numbers of a more suitable AMF might just as much economic benefit as large numbers of a less or underperforming AMF, if that makes sense.
Yep.
The reason that I analyze roots and soil is that because the symbiosis is so highly evolved and so it's so different among different plant families, even though mycorrhizal fungi are not host specific, so they're host promiscuous, we call it. They're very plastic environmentally, so they can move from host to host. And one plant can accommodate many different species of mycorrhizal fungi. However, the fungi are going to misreport to the plant what's available in the soil. It's in the fungus's best interest to hoard phosphorus. So even though we think of it as a symbiosis or a mutualism, it's important to remember the fungi for themselves and to think of it in capitalistic terms rather than egalitarian type terms is a lot more helpful. We use biological market theory to understand them in many scenarios. Fungi will hoard nutrients for themselves. They'll dole it out. They'll inflate the cost of phosphorus. They do things that look unnervingly like insider trading, and they'll short the market if you let them. And that's just how they play ball. Think of it more like a mutualistic antagonism than some kind of fair trade type thing. So the plant is going to, in response to these kind of tricks and games, is going to keep its root system under surveillance, and it's going to prematurely lyse any of those fungal symbionts that are underperforming. It's by this process that you get a selective enrichment of compatible fungi in the crop over time, which is why when I'm looking at fungi at the end of the crop cycle, I can see by tracking it over time how the host plant has kind of weeded out the underperforming fungi and ended up with those ones that are most conducive to plant needs. Now, remember, what plants want to do is they want to seed, right? And what fungi want are sugars and lipids, because they can't make lipids on their own and they don't photosynthesize. But we don't necessarily want plants to seed all costs. We want we want agronomic traits out of the plant. So we have to balance all these factors, and we have to look at which plant fungal pairings provide us with the best agronomic outcomes. There might be some environmental motivation if you're doing some soil remediation or trying to build your populations back up. But in sugarcane, the name of the game is yield, really. So you're looking to just improve yield per plant by a fraction. And then by virtue of having that kind of planting density and biomass, then that economic benefit actually becomes apparent pretty quickly. So I would encourage bringing more diversity into the cover cropping scenario and a mix perhaps, because you don't really know what's going to deliver the inoculum potential into your soil. Probably legumes, but legumes with a mix of cereals, as you mentioned, I think would be very good, because then you're not obviating those mycorrhizae that are better paired with cereals than they are with legumes, and they all interact. And like I said, there's a lot of competitive interactions even within the same host plant, where you can, even under the microscope, see these fungi just scrapping it out with each other. It's exciting, but systems on the brink can bounce back. So don't lose hope. Yeah.
So in all the sampling that we've done, and that's over a fairly diverse area and crops, 95% of the samples we get back, always 95% of the samples generally have detection of AMF A and B. Mm-hmm. We've never ever found anything with D, E, and only a very small amount of C.
Well, that's interesting data. I'd like to get my hands on that.
And that's obviously from northern New South Wales, southeast Queensland.
Yeah, I'm in Lismore, so.
They want heavy black vertisol clays.
Yeah.
And my concern is now, from your talk, obviously all these massive levels we're finding in the soil,
yeah,
they might be simply because the plants don't accept them. Is that what you're saying? So what we're thinking in the soil is good may actually be bad for the plant.
How long has the land been under cultivation with the same crop?
Oh, this is dryland broadacre farming, so 50 plus years of cereals more than likely.
Yeah, right. Good, because the fungi that are most conducive to that kind of abuse will proliferate. In sugarcane, we have land that's been under cultivation for a century or so, and it's in that land that we see selective enrichment of certain mycorrhizae, which have essentially adapted over time.
Yep.
They don't need evolutionary time to evolve. These things have a very high generation time. And mycorrhizal fungi they've got a very exotic biology, so they don't, even though we refer to them as species, their species definitions don't apply in any conventional sense. The fungi themselves harbor a whole range of nucleotypes, so genetically distinct nuclei that they can swap and change, turn on, turn off. Essentially, they become what the situation demands. And those fungi that are present in your soil, which would be, you said AMF-A and AMF-B, was it? So they're in there. So basically, next time you do a predictor test, ask Sadi to provide you with the additional DNA. Get in contact with the university or somebody who can organise that DNA to be sent for analysis, and I'd be very surprised if you don't end up with just 2 or 3 contenders. Once we know the species identification from within the assay, then we can just nail it and get right down and in there and understand, because heaps and heaps of work has been done on mycorrhizal fungi. It's just it's very heavily studied, and before too long, you'll find information about which fungi are present in your system and whether or not they are optimal from the amount of fungi you have. And you might even want to consider inoculation, but I wouldn't inoculate unless you were armed with this data first.
Excellent, thank you. So you've really got my mind spinning now, thinking about the site at Malali, which I just mentioned, where the grower had set up different levels effectively of AMF populations by planting the wheat, fallow and canola. So following the rain in February when it started, which broke the drought, we quickly jumped on the opportunity and planted some plots of barley to try and increase the AMF as one of our treatments in the trial.
Yeah.
We certainly did a fantastic job at doing that. Really build up those AMF populations of AMF B in that situation.
Yeah.
But my concern is now we think we've done a great job of building up the AMF B, but that might be absolutely totally useless for the sorghum, mung beans, and the dryland CoP, which is going to happen in summer.
Yeah, it might be that AMFB produce spores that contain thousands of nuclei, but don't produce many arbuskles in the roots of your crop, which would give you huge readings from predictor B and give you the impression that huge numbers of the organism exist in the system, when in fact very small numbers of the organism can exist in the system, and it's only by virtue that many nuclei are concentrated in the organism that you get large numbers. So I'm working, with Sadi now, so with Alan McKay and Danielle Giblot-Ducray down in Southern Australia, to figure out how we could translate kilocopies of DNA per unit of soil or root to the amount of organism that's present, and that's why I incorporate microscopy too. So one look at your roots in that system would give us a pretty fair idea of the amount of symbiosis that's going on, and it also gives an indication of how many mycorrhizal fungi that we're dealing with in the one plant. And you obviously, if the system itself has been starved of mycorrhiza, on the one hand you would expect there to be less colonization, but on the other hand, the plants might be under quite a degree of stress and might upregulate all those processes that recruit more and more mycorrhiza to help sort out the limitation. So you've got to kind of balance it's kind of one step forward, two steps back with a lot of this stuff, but eventually you get a clear picture of what's going on, and then looking at your spectrum of agronomic interventions. As far as your inoculum goes, then you could narrow down which ones might be suitable for use in your system, and then how you would measure their performance directly, because just going on what the label claims is just not good. I mean, it's like a wild west with that stuff. It's totally unregulated. And as I've demonstrated, just as a PhD student, raising the inoculum potential of your soil doesn't mean anything. I mean, the dominant fungi in the soil aren't even making it into the roots in sugarcane. If it's true for sugarcane, it's true for other crops, because I've just demonstrated, and if I need to do it across multiple systems, I've just put some ink in the water, you know. If it's true for mycorrhiza, then it's probably true for other soil microorganisms too. And mycorrhiza are only one component of the soil microbiological community, but why they're important is because they're the ones that draw down that carbon and put it in the soil. And it's those root exudates and those kind of residues that really foster a lot of microbial activity. So think of them as kind of soil health barometers. If they're suffering, everything's suffering.
That's fantastic, Anders. I love that that last sentence. It's brilliant. It's interesting how it's all everything's interrelated. You know, like everything affects everything, really. You know, and Brendan, in the chat, I've pasted the link to the Predictor B research. So perhaps you've been looking at the Predictor B services, but there is a service specifically for research. So perhaps if you go to that link and have a look, you can drill down and, like Anders was saying, be able to make those specific requests for your situation. So,
all good.
Thank you very much, Justine and Anders. Apologies for going over. I'll take responsibility for that because we started late. But thank you all very much for hanging in there. And if you have any more questions, I'm sure Justine and Anders would be happy to, on their behalf, just saying that they'd be happy to communicate with you guys, or if you want to communicate through me, I'm happy to do that and pass that on to Justine and Anders. Justine, we have had a request for a copy of your slides. Are you happy to share a copy of your slides for the group? Is that?
Yeah, that's possible. Yeah.
Yep. Thank you very much. So yeah, thank you everybody. Like I said, again, for hanging in there, and Justine and Anders for your time and commitment to this area. It's like we've learnt there's still a lot of unknowns, but it's great to have you two guys at the front there working away at it, and knowing that we can come to you if we've got any queries and for resources and that sort of thing. So
thanks for the opportunity to share my work. I appreciate you through all that.
yeah, great. Thank you.
And please give me a ring if you want to chat. I'll just put my email down if you want to ask anything specific. Very happy to chat about that, especially monitoring and showing extension tools for growers, getting them already excited about seeing soil biology in action.
Yeah, fantastic. Thank you very much. So thanks everybody. We'll leave it there, and I'll catch you all next week. And if you've got any queries in between, just send me some email or yeah, give me a call. So thanks very much.